So this is my attempt to try to stay on time here. Yes, so I want to thank you for the invitation and the opportunity to present to you. I am intending to give you some additional motivations beyond cancer to understand, in particular, the lung. So yes, I am coming to you as a physician scientist. I'm a neonatologist with career-long interests in postnatal lung development, injury, and repair. So when asked to talk about 3D tissue map data generation and value for biological questions, it was a foregone conclusion that I would be talking to you about the lung. It's my favorite organ, and you can't live without it. I'm going to be sharing with you the work, as Nils said, of multiple institutional collaborations. I'm PI for the Human Biomolecular Atlas lung component, DMC, and also for the Developmental Lung Molecular Atlas Program, or Lung Map, both funded by the NIH, the Common Fund, and NHLBI. I have no disclosures, unfortunately. So the 3D structure of the lung is critical to its function. The adult human lung, as you're sitting there, must accommodate the ebb and flow of about half a liter of air with each breath at rest. When you go out and exercise this afternoon, because we've been sitting here for so long, you're going to accommodate 3.5 to 4.5 liters of air with each breath. And you're keeping in reserve at least 1 liter between all of those breaths within your lung so that your blood can continue to pump and be oxygenated. This air must be warmed, humidified, filtered, and rapidly expelled with irritation or accumulation of secretions when you cough. And we accomplish much of that with the extensive branching airway structures that we'll talk more about here today. So your lung must also accommodate about 1/2 liter of blood at any one time, with increasing rapid exchanges with exercise to keep up with all that air. The flow of air, though, must be matched exactly to the flow of blood, or a mismatch occurs and the lung fails. In addition, the cells detoxify our environmental inhalants. They attract and return immune cells to the extra fluid back to the bloodstream, and they themselves must receive nutrition and be rid of metabolic waste. So we can identify and count at least 23 levels of branching airways in the lung. We can identify the structures and the cells responsible for gas exchange. And recently, we're able to discern up to about 80 different cell types in the lung. On the right-hand side, you can see much of the matching of blood flow to air occurs at the level of the gas-exchanging membrane of the alveolus, surrounded by capillaries, bringing in deoxygenated blue blood and putting out oxygenated red blood. But capillaries without aerated alveoli, or aerated alveoli without capillaries, represent dead space, or shunt, and inefficient breathing. There are systems in the lung that help to maintain that matching, and we'd need to understand them better to be able to take care of our patients, especially in the ICUs. On the left-hand side is a equation that describes the resistance to airflow. So the ability to move air through these tubular airway structures is directly, the resistance to it is directly proportional to the radius of the tubes to the 4th power, such that small changes in the size of these tubes have large effects on the flow of air and the work of breathing. Overall resistance decreases, though, as we proceed down the airway, despite smaller tubes, because there are increasing numbers of these tubes if they develop appropriately and are maintained. So it is important to be able to measure the diameter, the length, and the number of these branching airways throughout the lung. This is, of course, very difficult to do with 2D histology, and the promise of 3D lung tissue maps is to be able to make these measurements in healthy lung and compare it to disease. And unfortunately, things frequently go poorly in the human lung due to infection, environmental exposures, prematurity, autoimmune disease, asthma, and others. And as I said, I'm coming to you as a physician scientist, a neonatologist. I'm particularly aware and concerned about the 13 million babies that are born prematurely every year for the 37th week of gestation. Of these, about 15% or 2 million are at risk for significant chronic lung disease or bronchopulmonary dysplasia. And hopefully you can see there from their 2D, actually, chest x-rays, that this disease ranges from mild to severe cystic disease that carries morbidities that you can see on the left-hand side. Hypertension, susceptibility to infection, cardiac failure, premature aging, and a process of dysynapsis where the airway growth and development doesn't keep up with the lung growth and development. And this is a precursor to chronic obstructive pulmonary disease that we see in adults, whether or not they were born prematurely. The COPD occurs in about 10% of the population. Excuse me, maybe I have it. And is a frequent cause of early mortality. So one quick picture of 2D to 3D is that upper right-hand chest x-ray that you maybe can tell has too much air trapped in it. It's kind of cystic looking. If we go to a 3D CT image, of which I've only given you a 2D plane, but you can see how much more information we gather from that 3D reconstruction, as well as comparing it to the histology on the left-hand side. But again, in 2D, we lack a lot of information about why we have that obstruction in the airway. So for fun, let's take a journey through this reconstructed high-resolution CT scan of an adult lung. It'll take us about 30 seconds. You'll move about 8 liters of air while we're doing this. We'll go from the beginning of the airway down as far as we can reach, about 15 branches, to where we are limited by the resolution of the CT technology. I'd like you to notice that the walls are not smooth and that there are many dichotomous and orthogonal branch points. Each of these irregularities and branching nodes cause turbulence in the tidal flow of air, increasing resistance, but also facilitating filtering as particles fall out during turbulence and can be removed by the ciliary escalator. You can think also that there is pulsatile blood flow happening underneath this surface, further agitating the flow of air. But this is only structure. And what about the cellularity that composes the structure? So as you know, recent technological advances have allowed us to identify and catalog with increasing granularity the various cell types, subtypes, and states in the human body at the single cell or single nucleus transcriptomic level. With advances in imaging technologies that Melissa showed beautifully in their mass spec work, detecting now hundreds to thousands of RNA transcripts in tissue sections in situ, and with increasing protein detection by multiplexed antibody-mediated methods and high-resolution mass spectroscopy, we can now see the beautiful organization of these cell types, 50 to 100 per tissue, up to 1,000 in the brain. So here I'm showing you a view. It's just art. But it also tells us quite a lot. It's a view of the cells immediately below the tube that we just slid down, using just 15 antibodies. At the top, in the yellow-green, you're seeing the epithelial lining cells with cilia and mucus production. And in the purple, basal cells that are available as precursors to repopulate the epithelium. Those green, yellow, purple cells all look pretty uniform here in this 2D with only 15 antibodies. But I'll show you that they are not when we tease apart the many cell types that are present. Below that, you see bundles of smooth muscle, and there would be supportive collagen and other molecules of extracellular matrix. And embedded in the ECM, we have many structures and cells. The kind of round structure that you see in the lower left are actually submucosal gland ducts that end in submucosal glands that are producing mucus and self-defense molecules. Immune cells tend to hang out there as well. And there's also a mixture of epithelial cell types in those glands. Although this part of the lung is not involved in gas exchange between air and blood, it is highly vascular, as I hope you can see in the red CD31 or PCAM staining. Those circular red structures scattered throughout. T and B cells are quite numerous, as well as mast cells that are in turquoise there. You can see best down in the lower left. The lymphatic vessels are outlined in yellow. And maybe you can't see on this screen, but the nerve bundles are also visible, as well as scattered nerve fibers. So what biological questions of this area? And what is the normal include? What are the normal composition of these vessels and nerves? Where do they originate? Where do they lead? How are they interconnected? And exactly which and how are these immune cells trafficked? And how are they organized? Although we can learn much from these 2D images, they do not capture the cellular organization and spatial relationships needed to understand the intricate architecture and function. The promise, again, of a high-resolution 3D maps is to visualize and measure in multiple directions, count accurately, and observe in healthy tissue, and to see variations between humans and changes that occur with exposure and disease. So I'm going to give another example of the airway details that we're missing in 2D by looking at 2D. So Peter mentioned the kiwi, and the kiwi is actually very instructive. It's also delicious. And what you see in that kiwi is when we cut it across section, is like seeing the airways in cross section in 2D. And as, I don't know if this works or if it's worth, oh, it's not going to do that. Okay. So what you can see in the upper 3 panels and the first one on the left-hand side is by color, you can see different cells distributed throughout that airway that looks so homogeneous in that first picture that I showed you. But they're not. There are multiple cell types in there, each with its own function. Well, what I'm showing you here with the kiwi is if you cut it longitudinally and you section it down far enough, most of you are very good at visual orientation, so you'll understand what I mean. You'll get down to a point where you can see the bottom half of that tube, that airway that's moving through there. And the two images over to the right-hand side show you that. So here we're seeing now patches of these different cell types identified by color. And we can ask, again, new biological questions. Are these cells clonal? Are they interacting? Are they more likely to form cell type pods like this with disease, like asthma or smoke exposure? And are they like this only at those branch points, the nodes that I showed you in that CT scan, where the airflow is turbulent and there may be more damage to the epithelium? So these things we're hoping we can identify in 3D. So we have done some preliminary work in the lung with true 3D. These are done by a multiphoton microscope with cubic clearing and second harmonics generation. It's helpful to display this type of data in multiple formats, so such that we can interact with it and visualize different components. So I'm showing you 3 examples, actually, for the next 3 slides of the same thick section. Here as a serial Z-stack, you can see the blood vessel that is heading up to the right now and dividing into 2, and it's sitting right next to that round airway. Here we go back. across the view, and you can follow it through. So we can make measurements and understand relationships in this kind of image. Here, we're looking just at the connective tissue without the cellularity. I would love to be able to see the epithelial cells as it lies on that extracellular matrix, as well as the blood vessels running through them. And in this view, where certain types of nuclei are stained, we can start to understand the cellular composition of those structures, all on the same image data set. But depending on how you display that data and allow us to interact with it, we make different observations and new hypotheses. So in these next couple of slides, I'm going to focus on the alveolar septum. So that very thin membrane where gas exchange is happening. That alveolar structure is maintained by an extensive elastin fiber network that interconnects all portions of the lung. It's why we can expand our chest wall, lower our diaphragm, and open up our lung. It's all interconnected. And it's also why when we exhale effectively. The elastic recoil brings the lung back down and pushes out the air. So abnormalities in this network occur in diseases like COPD and BPD, and we'd like to be able to visualize them and measure them in healthy and then with disease. This shows the cellularity of that gas exchanging alveolar septum. It is very thin, has a capillary network shown in red, if you can see that. It accommodates single red blood cells flowing through it. And it's covered by alveolar epithelium, the green alveolar type 1 cells, and the cuboidal white type 2 cells. Type 2 cells produce surfactant, type 1 cells allow for the gas exchange. The elastin fibers that I showed you before fill up those dark holes at the end of each of those septal walls and form the cup, the upper cup-like rim of the alveolus. So this is one of the problems that we have. Looking closer at these cells composing the alveolar wall, the type 1 cells that you can see in yellow in both the drawings at the top and the bottom on the right-hand side, they are very attenuated and spread out. I think of them as a fried egg. I've got a nucleus, and then the white is just spread over the frying pan. They cover about 5,000 square microns on their basal surface. The endothelial cells, that capillary network is shown in pink, one endothelial cell in pink on the left-hand side, with a little blue nucleus, also is spread across multiple alveoli. So you can imagine, I think, as we take 2D sections and we cut through that type 2 cell, type 1 cell or type 2s, and we cut through the endothelial cells, and then we try to segment based on nuclei, that we're making very poor counts, very poor assumptions about which cell we're counting. We also end up, as others have said, getting blurring of the protein markers because they're so tightly opposed and overlapping each other. And so annotating those cell types becomes very difficult. We see multiple markers of different cell types that are hard to annotate or inaccurately annotated as a specific cell type. Okay, so I was also asked, what are we doing in RTMC to address 3D data production? You've seen some of our true 3D with a multiphoton. We will be moving to light sheet as well, but we have not generated very much of that data yet. And in the meantime, we're working on the serial section 3D data reconstruction. So we also have used tissue microarrays that you see there, 5 separate ones of them. These are very small. They're about 3 millimeter cores, but they allow us higher throughput at a lower cost, and will give us relatively small data sets. Will generate over, I'll tell you that in a second. So we are using autofluorescence, serial section H&E, and then multimodality, xenium, spectral stimulated Raman spectroscopy, and glycan by multi-mass spec, extracellular matrix, again by multi-mass spec, and then phenocycler with about 54 antibodies, followed by H&E. We can do that all on a single section. We've demonstrated that data, and that will be very informative as we integrate those modalities. Serial section for geomics to target on certain structures. H&E again, some additional MALDI and phenocycler, but then a set of about 30 serial sections. We've already cut these. They're just waiting in line now for phenocycler staining. There'll be 5 micron, about 25 to 30 of them. So covering about 125 micron total. That will give us most of the smaller vessels, at least once or twice, and the alveolar structure as well. Because of the limitation of the data coming out of each one of those cores, we also have section single whole slide images. And our whole slide images are about 1 centimeter by 1 centimeter or 1 by 2 centimeters, covering about 400,000, 500,000 cells at a time. And we'll run these through, again, multimodality, same section, same slide, as well as some serial. So last slide, advantages of the lung for 3D mapping. It is porous and it clears easily. Antibody penetration is a little bit easier than with other solid tissues. And I hope you gain some enthusiasm for me that there are very interesting structures, biologically and clinically relevant, with diverse cell types and functions. But what we need. So we do need multiplex methods in 3D to understand that cellular diversity and neighborhoods. Using 1 or 2 or 4 markers even is still quite limiting. High resolution to distinguish and trace the cells of the alveolar wall. So to be able to tell where a type 1 cell starts and ends, for example, and which nucleus belongs to which cell is necessary to really do this right. It is a repeating structure, but it's not compact. You can freeze the lung and it will collapse all down together, which is great because you don't have to image all that airspace, but it's terrible morphology. And so to try to, again, sort those cells one from another becomes very difficult. So we actually inflation fix our FFPE, which is why our histology looks a little, to my eye, better than much of the lung you see. But it requires multiple samples of multiple large regions to be able to reconstruct these structures. We need to be able to register and reconstruct 3D from 2D for various reasons. One, that we can generate that data more rapidly over larger areas than currently we can with true 3D microscopy. We need an ability to move those large amounts of data around rapidly. And I'm glad to see that there are some methods coming to do that. And of course, limitations in data storage and computing capacity. 20 terabyte scares me, cost-wise. We are generating well over 1,000 cores worth of phenocycler data over the next 3 months. Ability to interrogate the data, interact with it visually, perform cell counts and geometric measurements in a reproducible and automated manner so that we can test hypotheses. And then generating enough data in 3D to train and use machine learning. So I'm really glad that all of you are enthusiastic about doing this and you understand it better than I do. and can develop these algorithms. So great to work with you all. Thank you. And I have to acknowledge that this is a collaborative. We work with pathologists at University of Washington, our colleagues at UC San Diego, Pacific Northwest National Labs, the University of North Carolina, and my cores and colleagues at the University of Rochester. It takes quite a team. So thank you.
Thank you for this great presentation, Gloria. Very inspiring.